This is yet another video for my Biology for Health Science majors. We're still on Unit 3. This is the final Unit 3 video on nucleic acids. As we discussed in the last video, the amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. Genes are built of DNA. The word DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. DNA is inherited from an organism's parents. Your parents, for bacteria, they inherit from their parents as well. DNA is a message, a chemical message that's been passed from the very first living organism on the planet all the way through time to you today. DNA provides directions for its own replication. It also programs the entire cell's activities by directing the synthesis of proteins, which you learned in the last video. Proteins are the dynamic components of cells. They're the things that do everything, that get everything done for the cell. So DNA programs the cell's activities through proteins, and it also directs its own reproduction. Now, DNA is a huge molecule. It's the complete library, the whole thing. It codes for every single protein in the body. Now, this library is huge. It's bulky. We don't carry entire libraries to construction sites, do we? So the library has to stay in the nucleus of the cell. Bacteria don't have nuclei, but they still have little regions where the entire library is clustered. The DNA library is clustered. So DNA is not actually used directly to build proteins at the construction site, at the protein construction site. There's a middleman. So what happens is relevant sections of DNA are photocopied into a closely related nucleic acid called RNA. RNA stands for ribonucleic acid. So RNA photocopies of a gene are brought to the protein construction site and used as building blueprints. So the cell isn't making all the same proteins all the time. Maybe it needs to send out a communication signal, a protein communication signal to its neighboring cell. Maybe you need to produce more dopamine for the current situation. Now a particular section of the DNA has to be accessed. So that part of the DNA library is opened up. A photocopy of the relevant gene for that particular molecule you need to make in that particular moment, it's photocopied to RNA. The RNA goes to the place of the site of protein production. So DNA is transcribed into RNA in the cell's nucleus, in eukaryotic cells that have nuclei. RNA is translated into proteins in the cytoplasm of the cell, which is just, that's going to be just the liquid inside the cell. It's just like floating around in the cell's liquid. So here's a flow chart of how that all works. So you have DNA transcribed to RNA, which is just a photocopy of a small segment, just one gene. And that RNA, because it's small, it's more portable. So you can take it to the site of protein construction and use the instructions in the RNA photocopy to build that protein. So here is the monomer of DNA and RNA. So nucleic acids like DNA and RNA are composed of monomers called nucleotides, and this is one nucleotide. And each nucleotide has 3 parts. They each have a 5-carbon sugar and a phosphate group and a nitrogenous base. There are 4 different nitrogenous bases, and this is one of them. We have the 4 different nitrogenous bases are adenine, thymine, guanine, and cytosine. Those are the A's, T's, G's, and C's of the genetic code. So a nucleic acid polymer, a polynucleotide, forms from the nucleotide monomers when the phosphate group of one nucleotide bonds to the sugar of the next nucleotide by dehydration reactions. This produces a repeating sugar-phosphate backbone with the nitrogenous bases sticking out from the side. So just like with proteins, we had a repeating component that was the backbone, and then these non-repeating components that stick out the side. In this case, it's the nitrogenous bases. So here's a picture of that dehydration reaction. We have hydroxyl groups on these 5-carbon sugars. And then we have the phosphate group here, a hydroxyl group on this phosphate group. So again, you have H2O recombine and water leaves. And that allows this remaining oxygen to bond directly with the phosphorus in the phosphate group. All right. And so you get these repeating, it's sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the backbone of DNA. That's not the coding part. The sugar phosphate backbone doesn't contain any genetic information. It's just like the book binding. It's what holds the code together. And then these nitrogenous bases, you can see there's different nitrogenous bases. They stick out the side, and the sequence of nitrogenous bases is what provides the genetic code for building proteins. Now also notice that different nitrogenous bases are different sizes. So there's 4 different nitrogenous bases. Some have 2 rings, and some have 1 ring. And there are nitrogens in here. They're not hydrocarbons, despite the fact that it looks like that here. All right, so here's another picture of this repeating sugar phosphate backbone with the nitrogenous bases sticking out the side. Different nitrogenous bases are different sizes. And the sequence of nitrogenous bases is what creates the code for polypeptides. So this is a 4-letter language. Computers use a 2-symbol language, 0s and 1s, and DNA has a 4-symbol language. Now, just like proteins had a C terminus and an N terminus, giving them some kind of directionality, RNA and DNA also have directionality. So there's, it's the sugar phosphate, sugar phosphate, sugar phosphate repeating backbone. The end that ends with a sugar is called the 3' end, and the end that ends with a phosphate is called the 5' end. And I'll explain why. We can number the carbons in the sugar, and when we number them, we move around clockwise from the oxygen. So, because sometimes, we draw the molecule in different directions. So you find the oxygen in the ring and you move clockwise from that. So this is carbon 1, carbon 2. Remember, the corners that are unlabeled are carbons. Carbon 3, carbon 4, carbon 5. All right. So 1, 2, 3, 4, 5. And in the chain, in a single nucleic acid chain, all the sugars, you'll notice, are oriented in the same direction. So that if this 5th carbon is pointing up in this one, all of the 5th carbons are pointing up. So 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So the 5th carbon is always pointing towards the phosphate group, and the 3rd carbon, this should be 3, sorry. The 3rd carbon is always facing away from the phosphate group. So in this nucleic acid chain, the sugar end, the end that terminates in a sugar, that's going to be called the 3' end, because that's the direction that the 3rd carbon is facing. And the end that terminates in a phosphate group, that's going to be the 5' end, because that's the direction that the 5th carbons are all facing. Now, keeping track of directionality of a DNA and RNA molecule is actually really important. We have cracked the genetic code. When we look at a gene, we can decipher it and figure out what protein is produced. But the directionality matters because the enzymes that read the DNA and build a protein from it, they always move in the same direction along the gene. Sort of like you always read a book in the same direction. If you turn your book around and look at it through a mirror so that all the words are backwards, it doesn't make any sense. The words only mean something in one direction. They don't mean the same thing backwards. They probably mean nothing when you read them backwards. So in the same way, it matters which direction you're reading a DNA molecule. So if you want to figure out what protein is produced, you have to read the DNA in the same direction that the enzyme is going to read it. So you need to be able to keep track of which end is which. Where is the 3' end and where is the 5' end? Now, another thing I want to point out is that this is DNA on the left and this is RNA on the right. And how do I know? DNA is missing a hydroxyl group. It's deoxyribonucleic acid. That means it's deoxygenated. So this 5-carbon sugar is ribose, But in DNA, we have deoxyribose, deoxygenated. It's missing a hydroxyl group, versus these ones have that hydroxyl group there. Now, there are consequences to this. This is a very, subtle difference, of course. So DNA and RNA are almost exactly the same molecule. But remember, hydroxyl groups are quite reactive, which means that RNA tends to react more readily with things, with other chemicals in the environment, and that causes it to break down more quickly. Now, there's tons of RNA left at crime scenes, but we don't use it, in part because it doesn't last as long. It's not as stable a molecule. For the same reason, we don't look at RNA when we dig up a woolly mammoth or a caveman or something. We're always looking at DNA because DNA is more stable over time because it doesn't have those reactive hydroxyl groups. It's deoxygenated. Another thing I want to point out about the difference between RNA and DNA is the nitrogenous bases. So here's, a bunch of nitrogenous bases sticking out the side, and here you can more clearly see that there's nitrogens in every single one of them, which is why they're called nitrogenous bases. But these are mostly the same as each other. There's one difference between this one and this one. So in DNA, we have thymine, and in RNA, we have uracil. The difference is this methyl group sticking off the side. So again, you don't have to memorize the chemistry of that. I'm just telling you that one of the letters, the chemical letters, is different between DNA and RNA. Now, DNA has the shape of a double helix. And remember, again, helix is a spiral shape. Because two chains wind around each other, that's why we call it a double helix. And these two chains are held together by hydrogen bonds between them, right? You can see that those dotted lines are hydrogen bonds because the nitrogenous bases are polar. So again, you see these sugar phosphate backbones running up the sides in blue and yellow, and the nitrogenous bases stick out in between and they meet up with each other like rungs of a ladder. So the shape of DNA is sort of like if you took a ladder and twisted it to look like a spiral. Now, there are rules for which nitrogenous bases can bind to which. C's and G's always bind together, and A's and T's always bind together. You're not going to find A's binding to G's or T's to G's. It's always A to T and G to C. Now, these are called complementary base pairing rules, and the basis for the complementary base pairing rules is chemistry. The number of hydrogen bonds and the orientation of those hydrogen bonds will dictate which nitrogenous base can bind to which. Adenine and thymine each can form 2 hydrogen bonds. Guanine and cytosine each can form 3 hydrogen bonds. So guanine and cytosine will always bind together. Adenine and thymine will always bind together. This means that the 2 strands in a DNA molecule are not mirror images of each other. They are not identical, but they are complementary, meaning if I know the sequence of one strand, I can infer the sequence of its complementary strand. Because everywhere I find an A over here, I know I'm going to find a T over here because of the complementary base pairing rules. All right, so let's summarize the differences between DNA and RNA. We've already seen how they're almost identical molecules. So how do they differ? Well, in DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. The difference in the name is because of the difference in the 5-carbon sugar. So in DNA, we had deoxyribose, and in RNA, the sugar was ribose. They're both 5-carbon sugars. The difference is in that one hydroxyl group. Ribose is a little bit more reactive, and deoxyribose is a little bit more stable. So that makes DNA a more stable molecule than RNA. Also, DNA is extremely long. There's 6 feet of DNA in every single one of your cells. If you were to unwind it and stretch it out, it would be too narrow to see, but it would be 6 feet long. So that's because it's the entire genetic library for the entire living organism, while RNA is very short. It's the length of a single gene. DNA is double-stranded. It has that double helix shape. RNA is single-stranded. And there's the difference in that single chemical letter. The chemical letters for DNA are A, T, G, C. Adenine, thymine, guanine, cytosine. In RNA, we replace thymine with uracil. So it's AUGC or adenine, uracil guanine cytosine.